Hi, so we're going to continue the macromolecules series with nucleic acids, and we're starting with DNA. So you probably know that DNA is the instruction manual that gives us all the traits that we have as carried out by RNA and then proteins. DNA comes in our cells in the form of chromosomes. We have 46, 23 from each parent. In eukaryotes, which are us, animals, plants, fungi, we have nuclei. So we have a membrane-bound nucleus in our cell where the chromosomes reside, and they get to be protected from all the DNases and other things in the cell that might degrade them. The other thing about eukaryotic chromosomes is that they are linear. Meanwhile, in prokaryotes, bacteria and archaea that do not have a nucleus, they have only one chromosome. It is in a circle, and it resides in an area of the cell known as the nucleoid. So that's kind of like the nucleus, but there's no membrane around it. It's just an area. So why do we care about DNA? Like I said, it's the instructions, and DNA is going to go through a process called transcription, in which DNA is going to serve as a template for RNA, and then RNA will go through a process called translation to turn it into a protein. And that protein is going to do everything that the gene told it to do in the beginning. And that whole process, we call it the central dogma of biology because it is probably the most important thing in molecular bio. So what does DNA look like? On the left here, you can see the structure of one nucleotide. So all these biomolecules that we're talking about are polymers, so they have a lot of repeating units, and the repeating unit in DNA is a nucleotide. This nucleotide has got three parts. You've got a sugar in green, a phosphate group in pink, and a nitrogenous base in blue. The nitrogenous bases are what make our DNA sequence our DNA sequence. The order of the nitrogenous bases is going to determine the order of amino acids in the protein. There are 4 different nitrogenous bases. There's adenine, thymine, guanine, and cytosine. And we can categorize them a few different ways. So adenine and guanine are purines, which means that they are big and have 2 rings. Meanwhile, thymine and cytosine are pyrimidines. They have 1 ring and they're littler. Now, because we want DNA to have a uniform diameter and not to go like fat, thin, we want to pair 1 purine with 1 pyrimidine always. So adenine is going to pair with thymine and guanine is going to pair with cytosine. Well, why doesn't adenine pair with cytosine? The reason for that is that the nitrogenous bases in double-stranded DNA are connected by hydrogen bonding. Adenine and thymine each make 2 hydrogen bonds, while guanine and cytosine have the capacity to make 3 hydrogen bonds. So the hydrogen bonding, along with the uniform diameter, is what determines the base pairing of adenine, thymine, and guanine, cytosine. As you go on in biology, you're going to find that in the lab, when we want to make a stable DNA, we want it to have a lot of GC content because that's going to give it a higher boiling point because there's more hydrogen bonding. The other cool thing we can do with base pairing is we can predict the percentages of one base based on another base. And that's through something called Chargaff's rules. So Chargaff's rules basically say what we just said, that adenine and thymine pair and guanine and cytosine pair. So, for example, if I know that we have 30% A, then by default, we also have to have 30% of T, right? Because if adenine pairs with thymine, we need the same amount of A and T. So now we know that A and T compose 60% of this hypothetical DNA. So that leaves us with 40% left for the other base pair, G and C. So we know we have to have the same amount of G as C, so we just divide 40 in 2 and we get 20% of each. And this is a classic problem, and you'll probably see it on a test if you're doing a unit like this. So how does DNA come together? If you look on the left side, ignore all the DNA replication stuff for now, you're going to have repeating units of the sugar and phosphate of the nucleotide in a sugar-phosphate backbone. Those are going to be connected by what we call phosphodiester bonds. if you study chemistry, like basic organic chemistry, you're going to learn about the functional group called an ester, which looks like this. And the ester group is this. And a phosphodiester is just going to be like a fancy ester using a phosphate. So you see how here the phosphate has got 2 of these oxygens. This one is going to lead to another sugar, so it's like a diester because it's got 2 extra things. So that's the sugar phosphate backbone, and like I said before, the bases are going to be linked by hydrogen bonding. So remember that covalent bonds are stronger than intermolecular forces like hydrogen bonding. So it's going to be easy for the DNA to unzip down the middle, and the backbone of the sugar phosphate is still going to remain intact. And that's how DNA replicates. It replicates through something called the semi-conservative model. So the DNA is going to unzip through the hydrogen bonds at the bases, and it's going to turn into 2 separate strands of single-stranded DNA that just have 1 base all the way down through the nucleotides. Now, because of the base pairing rules, the enzymes in the nucleus can go ahead and make a new strand of DNA that looks just like the one that came off based on the base pairing rules. Now, this replication process is kind of complicated. It's got a whole lot of steps and a whole lot of enzymes, so we are going to go through it pretty slowly, and we're going to try to cover it all. So, DNA is going to unzip, like we said, and this unzipping of these 2 strands is going to be done through an enzyme called helicase. And helicase is going to do this at a lot of different places in the DNA. In our linear chromosomes, they're really long on a molecular level. So what ends up happening is we open our DNA strands in lots of different little places called replication, origins of replication. And they're going to make little replication bubbles. And these bubbles are going to expand as the DNA replicates, and eventually they're going to fuse, and then you're going to have a full new 2 daughter strands. So if we zoom in on the replication bubble, if we divide the bubble in half, 1/2 of that is called the replication fork. So at this replication fork, where the DNA strands start to divide, is DNA helicase unzipping them. Now the unzipping of DNA is a little bit problematic because it creates this thing on the other side here where it's still double-stranded called supercoiling. So supercoiling is exactly what it sounds like. It's just the DNA gets really coiled up and messy. And the way that our cell fixes this is an enzyme called topoisomerase. And topoisomerase is just going to make a cut in the double-stranded DNA, and it's going to let it untwist, and then it's going to put it back together. And it does this however many times it needs to while the replication is still in progress. The enzyme that actually carries out the replication is called DNA polymerase. Like polymer, DNA polymerase, that can be your way to remember it. And DNA polymerase is always going to move in the 5' to 3' direction on the DNA. Now what that means is, if you look back up at the nucleotide, this the nitrogenous base, we count the carbons 1, 2, 3. So on the sugar, we have to have another system, and we count them like 1', 2', 3'. So here's 1', 2', oops, not there, 2', 3', 4', and 5' is in here. So the 5' is attached to the phosphate, and the 3' will be attached to another one. And what ends up happening is these strands run anti-parallel, which means that at one end you have a phosphate at the end, and that we call 5' because the phosphate is attached to the 5' carbon of the sugar. And if we have a 5' end here with a phosphate, the other one is going to be a 3' end, and that's because the 3' OH of the sugar does not have a phosphate on it. And then at the other end, it's the reverse. So 3' and 5' here. But the point is that DNA polymerase can only go in the 5' to 3' direction. The problem with that is that we want to move consistently in one direction. So say we are moving this way and we are opening up the strands this way. Well, this polymerase is fine, right? He can just move along. But this one would be moving in the opposite direction. And that's kind of complicated. So what ends up happening is that the lagging strand, the one where it's going in the wrong direction, is going to make little fragments. So it goes a little bit this way, and then it comes back. And it goes a little bit this way, and then it comes back. And it keeps doing that. And so it's moving on a micro level, 5' to 3', but overall 3' to 5' to match the direction of the leading strand. We call these fragments Okazaki fragments after the dude who discovered them. And DNA polymerase does those in the lagging strand, and the leading strand is pretty simple. The other problem with DNA polymerase is that it can't start its own strand. It kind of has to have a little boost before it can work. And that boost is provided by an enzyme called DNA primase. And DNA primase actually makes a primer, a little short strand of new nucleic acid, but it's not DNA. It's going to be made of RNA. So this DNA primase is going to make a short little primer of RNA. And now that there's something to work with, DNA polymerase can start to do its thing. Then eventually a different DNA polymerase at the end is going to come back and replace this RNA with DNA. And then finally an enzyme called DNA ligase is going to come in, and it's going to put these two DNA pieces together. The other thing that's important in DNA replication is these little red guys. They're called single-strand binding proteins because you're going to open up the replication bubble and you're going to have these two single strands of DNA, and before polymerase comes along and does its thing, they're both single-stranded. What's to stop them from just going and sticking to each other again? That's the job of the single-stranded binding proteins. So that's replication up until we get to the end. And polymerase has another problem, because polymerase apparently has all the problems. It gets to the end, and it's like, I can't do this anymore. I can't finish this last one nucleotide at the 3' end. So eventually, as it continues to replicate DNA, like cell cycle after cell cycle, this DNA is going to get shorter and shorter by one nucleotide because polymerase can't finish that last nucleotide. So that's a problem because we're losing genetic material. It's actually been associated with aging. So the fix that our cell has for this is an enzyme called telomerase. It's going to come along, and telomerase is going to make this kind of overhang thingy called a telomere. And it's going to be just a bunch of nucleotides at the end so that DNA polymerase can replicate the end. Over time, telomeres are going to get shorter as we age, and telomerase is not going to work as effectively, but it works well enough for us to replicate our DNA and for us to keep our genes intact. And that's the important part. So that's DNA. I know it's a lot, but if I erase all my crazy drawing stuff, then maybe you can take a screenshot of this page or copy down things. When I was learning this, my textbook had a big table with all the enzymes and what they did, and I sat down and I studied that. So that's my suggestion for you. So now we're going to move on to RNA. So RNA is ribonucleic acid. DNA was deoxyribonucleic acid, and the deoxy meant that there was no OH on this carbon in the sugar, this 2' carbon. In ribonucleic acid, without the deoxy, it does have the oxy. It does have the OH. So that's the difference in the sugar between DNA and RNA, is this 2' OH is present in RNA and it's not present in DNA. The other difference is that RNA is single-stranded. Where DNA was this double helix thingy, RNA is going to be single-stranded. It's going to just be floating around as not a line, but kind of a curlicue, because what's going to happen is there's going to be random base pairing within the strand. So say this is my RNA strand, and there's like a C here and a G here. Suddenly this can pair, and then you have a loop thing. But other than that, there's not really any secondary structure, no 3D structure. It's just a single strand. Third difference is in the bases. RNA has A and G and C, but it does not have T. RNA has something else. It's different by like only one methyl group, but it's called uracil. So it's going to be AGCU instead of AGCT. So whenever you're on a test and you're being asked about DNA, if it's U, it's wrong. If you're being asked about RNA, if it's T, it's wrong. So what does RNA actually do? RNA is this intermediate step between DNA and protein. So remember our central dogma, we have DNA, we have RNA, and we have protein. So DNA and RNA are related in the bases. So when DNA gets transcribed into RNA, it's based on the base pairing rules. So you're going to get an RNA that looks exactly like the template DNA strand, or the coding DNA strand, except for the T's and U's. And then it's going to go to protein via something called the genetic code. So every 3 nucleotides or bases on the RNA is going to represent 1 amino acid in the protein. And when translation happens, you've got 3 different types of RNA working together. mRNA, or messenger RNA, is that transcript that we were talking about that matches the DNA through base pairing. Ribosomal RNA makes up the ribosome, and the ribosome is just this little unit that brings all the ingredients together that are needed for protein synthesis, and it's a complex of RNA and protein, and that RNA is called ribosomal RNA. It looks kind of like a hamburger. It's got 2 different subunits, large subunit and a small subunit, and between those subunits, the messenger RNA will sit and be read so that it can be translated into amino acids. The transfer RNA's job is to bring over the amino acid and kind of assemble them into a chain. So you can see the transfer RNA's got kind of like a clover shape, and it's got base pairing within itself in certain places. And it's going to have one end that is complementary to the messenger RNA with a specific sequence called a codon. That's those 3 nucleotides we were talking about. And at the other end, it has a sequence that's going to attract a specific amino acid. So, for example, if the codon here is A, U, G, that's like the nucleotide sequence on the messenger RNA, then this transfer RNA would say U, A, C, and because those are complementary, they're going to bind with each other through hydrogen bonding. Now, the AUG codon is specific to the amino acid methionine, so this would be methionine, and it would be attracted via intermolecular forces to this end of the transfer RNA. We'll have more on that later. I think I'm going to do a whole thing on transcription and translation, but this is what you need to know about the different types of RNA.